All right. Hello, everyone, and welcome to today's keynote presentation titled How to Grow Almost Anything, a robotics-enabled learning model for global synthetic biology education. This webinar is a part of the 8th annual event in the Laboratory Automation Virtual Event Series. I am Sydney McNeil of LabRoots, and I will be your moderator for today's event. Today's web seminar is presented by LabRoots. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the bottom left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many questions as we have time for at the end of the presentation. If you happen to have any trouble seeing or hearing the presentation, click on the Help Desk button from the lobby within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits image located on the right of your screen and follow the process to obtain your credits. Now let's get started. I now present today's speaker, David Sun Kong, synthetic biologist, director, MIT's Media Lab, Community Biotechnology Initiative. For a complete biography on our speaker, please find it on the left of your screen. David, you may now begin your presentation.
Okay, thank you so much. And it's a huge honor and privilege to be here with you all. I am going to start sharing my screen. Let me just give me a moment here. Okay, hopefully you are all seeing a presentation. Unless anybody says otherwise, I'm going to assume that this is working. So anyways, again, my name is David Sun Kong. I direct the Community Biotechnology Initiative at the Media Lab at MIT. Today, I'm going to talk about a global synthetic biology course we've been teaching called How to Grow Almost Anything, and in particular, how we've been using robotics to enable a new model for hybrid and distance learning education that I'm really, really excited about. So just briefly, I'm from the Media Lab at MIT, and for those of you who don't know, the lab I think is very special in that it explores a number of different domains of creativity, and you'll see the lenses of these creative aspects on the work that I share today. So not just science and engineering, but also including art and design. And that's a big part of what's reflected in the course I teach, How to Grow Almost Anything, and also in the larger, in a way, creative platform, creative engine that we're trying to build and enable with robotics. So one other very special thing about the Media Lab, we have a really great history of taking different types of transformative learning experiences that connect to up-to-date and futuristic technologies. So starting, for example, with coding and computation. This is the work of Professor Mitch Resnick, who runs the Lifelong Kindergarten Group at the Media Lab. Professor Neil Gershenfeld, who's been a pioneer in digital fabrication and teaches the course How to Make Almost Anything. And in my work in bio, the life sciences and synthetic biology. And in all of these cases, there's been some type of a learning experience that's been developed and then propagated globally through a network of some kind. So with Scratch and Computer Club, with Scratch and coding, the Computer Clubhouse Network, with digital fabrication, the course How to Make Almost Anything, which I'll talk about in a second, and a network of what are called Fab Labs. And then through my work propagating creativity around bio through a network of community biology labs, and then also this course, How to Grow Almost Anything. And so How to Grow came in a way very much inspired by Professor Gershenfeld's course, How to Make Almost Anything, which was all about digital fabrication, and is structured and modeled in a similar type of format where each week in the course, students are learning very particular skills that connect to big themes in digital fabrication. And How to Grow Almost Anything, and that is the Jurassic Park soundtrack you're hearing in the background. We take a similar approach, except for the life sciences, synthetic biology, and biotechnology. And this is a course that I've been really privileged to teach with Professor George Church from Harvard, Professor Joe Jacobson from the Media Lab at MIT, and many other faculty around the world. And so I'll go through the curriculum in great detail in a moment, but at a high level, again, each week we're learning some type of skill at the frontiers of synthetic biology. If it's about DNA design and editing, protein and artificial intelligence, machine learning, biofabrication, whatever those topics might be. And after about 13, 14, 15 weeks of this curriculum, you can hopefully, quote unquote, grow almost anything. And so the course, again, is taught by myself, George, Joe, and then so many pioneers and leaders at the frontiers of synthetic biology as a domain. So, oh, sorry, I'm just moving this. There we go. Give me one moment here. And so many other leaders in the field. Many of the colleagues shown here, folks like Emily LaProuse from Twist Biosciences, Drew Randy from Stanford, Patrick Boyle from Ginkgo Bioworks, and so many other amazing faculty from all around the world. And the other really special thing about How to Grow, the students don't actually have to have a previous background in biology, which is really, for us, very, very exciting and also very, very Media Lab. So many of the students come from other backgrounds like art, design, computer science, mechanical engineering, material science, urban planning, management, business, and folks that are just very, very curious about what's happening at the frontiers of synthetic biology and want to build hands-on skills. And so these folks, again, the majority have never been in a wet lab before. And so that's one aspect that's very interesting. The other is that we have, in addition to our MIT and Harvard students, we also have a global classroom, a virtual classroom. And this is, again, where we start getting into some of the robotics-enabled and distance aspects of the course. So, for example, in 2022, we had more than 300 students, accepted students from around the world. In 2023, more than 400. And so it's been growing really significantly every year, again, with students from all around the world, including here in Cambridge at MIT and Harvard. And for the students that have never had a biology background before, have never been in a wet lab, we start with a bio bootcamp. So this is where we teach students the very fundamentals of, what a DNA molecule is, what the central dogma of biology is, how to pipette, all of these basic skills we start with in this bootcamp. And then we kick off the course, the first official week, really focus on principles and practices. So how do we design? Why do we design with synthetic biology? Why do we want to build these systems? And how do we do it safely? How do we do it securely? How do we do it ethically? And this aspect of principles and practices is something that we propagate as a core set of values throughout the course. In week number 2, we talk about synthesis and sequencing and editing of life, in particular DNA. So it's a huge honor to have always George Church, Joe Jacobson, and Emily LaProuse, all three pioneers in DNA synthesis, DNA editing, and DNA reading technologies. This is Charles Darwin, and this is George Church. So George is like the chiral Charles Darwin. We then have a class on lab automation and cloud biology. A big part, again, of where I think the future of this course is headed is very much going to be mediated using robotics and automation. And then we do a class on protein design. So we talk about the fundamentals of protein structure, how to design them, what the applications might be. And this year, for the first time, we actually add an additional class, in part because of all the incredible advances that have been unfolding in artificial intelligence and machine learning connected to protein design. And again, I'll connect this back to robotics towards the end of the talk, but I think these types of developments are part of what we incorporate into the course as it evolves each year. So depending what's happening out in the world, we'll bring in different types of emphasis on other technologies that are really enabling the present and future of synthetic biology. Next, we have a class on bioproduction. And so here, typically we'll have Ginkgo Bioworks, again, a leading synthetic biology company in engineering organisms. We have them come be a part of the class. We actually just had class yesterday on cell-free systems. So Kate, Peter, and Ali came. And this is one of my favorite classes. Cell-free systems, I think, are such an enabling core technology for the future of synbio and biotech. And this particular class, we teach through the lens of synthetic minimal cells, so kind of origins of life, which is Kate's area of expertise. Peter then talks about how to use cell-free systems in the context of textiles, materials, for example, how to make masks or other lab coats that have freeze-dry cell-free systems on them that could be used for diagnostics. Ali Huang from BioBits talks about the application of cell-free systems to education and learning. We then have, there's 2 wildcard weeks where, depending on what's happening in the world, while folks talk about tissue engineering, astrobiology, biomaterials, RNA technologies, measurement and imaging, DNA nanostructures, whatever might be topics of interest that year. We always have a class on building genomes. So this is, again, we talk about building DNA of sizes from genes to genetic circuits, but then really also how to build minimal genomes of bacteria all the way up to yeast and even larger, visions towards even human genome construction. I helped to teach a class on microbiome and science engineering, so thinking about how to use the microbiome and the microbiota of the body and the environment in interesting and exciting ways. We also have a class on tools. So here we focus on microfluidics and also this area called frugal science, which has been pioneered by my colleague Manu Prakash. So using low cost, readily accessible technologies for global health applications. We have another wildcard week. And then we close with a class focused on biodesign and fabrication. So again, I think going back to my earlier slide, looking at art and science and design and engineering, designers are now really, and artists are getting into synthetic biology. And so it's really exciting to explore those frontiers in this class as well. And so after all of these, or during these weeks, as the students are getting this exposure to what's at the cutting edge of synthetic biology, then there's hands-on lab skills development, from looking at DNA design to working with Opentrons automation, to designing proteins, actually doing bioproduction, engineering of organisms to produce molecules like lycopene, beta carotene in class. Again, the students right now are working on a cell-free systems lab. Being able to build bigger pieces of DNA using technologies like Gibson assembly, working, of course, with CRISPR, which is a very exciting genome editing technology. Culminating in a final project. So this is one of, again, I think the big pedagogies that we have at the Media Lab. Students learn the best according to the 4 Ps of learning. This is from my colleague, Mitch Resnick at MIT. So with peers, play, passion, and projects. And so one of the huge parts that we believe in here in How to Grow as well is that the students learn the best when they're, and I think this is almost self-evidently true, you learn the best when you're doing things that you care about, right? When you have a passion, when you have an interest, that's when you do the most creative and dedicated and passionate work. And so the students, as they learn all these skills, then apply those skills at the end of the semester towards a project of their creation that they care about. So, and through this process, we get amazing projects. So for example, Genique Goldstein, she's a designer. She's very, very interested in textiles. So she used for her project an incorporation of engineered organisms that are responsive to different stimuli to produce different types of colors, and integrated those with her textiles. Rosalie Lin, another designer from Harvard, she made a bacteria screen printing fabric and collaborated with Miana Smith, who is a digital fabrication expert, on making a whole spinner system, excuse me. There's also computational projects, Matthew making this PETase enzyme for breaking down different plastics in the environment. Spider silk spacesuits, exotic luxury synbio. This is a project from Gomi Gomez from Mexico. He was imagining a future where you might use synthetic biology to create fantastical animals, for example, things like rare tusks or materials of endangered species to try to eliminate animal poaching. So all of those types of projects, this is another neat one from Laura Gonzalez, focused on architectural structures and bacterial shading at architectural scales, biosensitive tattoos, or handbooks for creating biological art. And this is kind of a neat story. Yishao Zhang, she had never been in a wet lab before, was one of our global students, and after the course became the CEO of her own biotech company. She was manufacturing and designing desktop biology labs. Another neat project, this is from Pat Bhattacharya from the Media Lab. His final project was on wearables that could be used in outer space. So could you imagine having optogenetics enabling the production of different types of molecules that might be useful for space exploration? He then tested his prototype in 0G. Also, you had this be, this was a big part of his own master's thesis. A.L. Perry did a project on DNA canvases that, again, was a big part of his master's and later PhD work. Belen was a management student, a business school student at Sloan School at MIT, and worked on a whole cell COVID-19 biosensor, which she then collaborated with colleagues at the JCVI to run prototypes and test. I love this particular story too. So this is Andreas Rico. Andreas is a city scientist. So he's somebody that cares a lot about the future of cities. And when he took the course, he was really inspired by thinking about how to enable cities with biosensors, and built this modular open source bioreactor. And that became a big part of his master's thesis. And then we just submitted a paper on his work. So kind of it turned into a thesis and then a publication. And it didn't end there because the whole project was open sourced. Corinne Takara, she's a current student in How to Grow Almost Anything and a designer from Hawaii. She took that open source design and made her own version of it, except to explore ancestral cultural knowledge connected to indigenous species in Hawaii. And now, kind of dive more deeply into the robots. So during the pandemic, because the students suddenly couldn't be in wet labs, we were faced with a real conundrum as an instructor team, right? Like, what do you do if students that need to participate in this hands-on wet lab course suddenly can't do wet lab biology? And so we came up with a really creative solution. We had the students program liquid handling robots. We taught them how to program Python, and they used robots that we had at MIT. And so we would have, for example, a TA in the lab, making sure the robot was running okay, but then students could be anywhere, right? The students could be across the street in their dorm rooms. They could be anywhere around the world. All you need is an internet connection and a computer, and all of a sudden you can access and engage in our learning curriculum. And so as an example, the bioproduction class, where we focus on lycopene and beta carotene production, in this particular course, we took all of the elements that were part of that lab. You've got a shaker, there's an incubator, and so on. And we set it up inside the robot, and we're able to execute the experiment completely remotely. And so this led us towards this vision that we've been incredibly excited about around using cloud programming, right? So could you then have a system where, using open source robots that could be distributed around the world or at MIT, you can have kind of a positive feedback cycle where folks are generating protocols, executing those protocols on the robots, getting data back, and then learning and developing creative skills as a consequence. And so this was a vision that we published in a Nature Biotechnology paper that came out about a year ago, entitled How to Grow Almost Anything, a Hybrid Distance Learning Model for Global Laboratory-Based Synthetic Biology Education. And the whole vision here, again, is about dramatically reducing the barriers of access to quality, transformative synthetic biology learning and education experiences, right? So can we take designers, artists, scientists, engineers from all kinds of disciplines and get them into SynBio by thinking about the ways in which we can use robotics to enable that type of hybrid and distance learning? And so as a part of this work, we took our curriculum and we figured out which parts of the curriculum could be done entirely with robots, which parts could be done TA assisted, which parts could be done in silico, just with your laptop. And then we basically structured our curriculum in that way. And so, you can do things like this. This is George Church's cells being actuated under experimental conditions designed by our students. So this is a distance experiment. And the other really cool thing about this particular vision, this is all again about this teaching practice in the context of global learning. All of the students in the class from those particular years were actually authors on the paper. And so, again, part of what's kind of amazing, a good number of the students who had not been in a wet lab before, after going through the course and participating in the work to make the paper real, suddenly found themselves co-authors on a Nature Biotechnology paper. And this is also true for our global students. Again, many of them had not been in wet labs before, were new to synthetic biology, and a year later suddenly found themselves co-authors on a Nature paper. So this was really exciting, and I think, again, points towards a vision of real collaborative research. And this is one of our bigger goals too. We love the idea of having students and learners from all around the world being able to connect and learn and do research together as a part of this vision. And so, again, thinking about the scale of the course, I mentioned, we had 300 plus accepted students in 2022, 420 in 2023. This year we're up to 545 accepted students from around the world. So we're really seeing incredible growth for the class. And now there are different chapters that are being organized. So this year there's an Asia and Oceania chapter. Again, the part of what's cool about the course, we do it live, of course, in a classroom, but there's also a Zoom classroom that's happening at the same time. So there's this whole parallel classroom where there are TAs from around the world via Zoom working with our global students. And there's just this, kind of the chat is always on fire with, people talking, engaging with each other in parallel, while the students in the room are having our own class. And of course, there's some back and forth between the online and the virtual and the in-person. And for students, for example, in Asia, it's like 3 a.m. for them. So they then do their own version of the course at a time that's better for them. They can then watch the lectures together and then do the homeworks together as well. And so these different regional chapters have been something that's been springing up for the past couple of years. And the other thing to mention that I'm really proud of with the course, the students after they take the course, they're all invited back to become mentors the following year. And so just as an example, this year in 2024, we have 60 teaching assistants as a part of the class. And they're from all around the world. 20 of them are from MIT and Harvard, and 40 of them are global. And the teaching assistants, 50 out of 60 of them were all previous students. And all of the global TAs were all previous students. So we basically set up this amazing kind of virtuous learning model where, once you take the course, you become a mentor, and then you're basically a part of this learning community that can grow out, over time, and it only becomes more robust and more creative, and more connected, and more impactful as time goes on. And so this for us, I think, has been a really exciting, global vision, thinking about how to do this type of design and synthetic biology work, but at a transformative global scale, and enabling it using these virtual tools, tools like we know, like Zoom, but then also tools like robots. And so this year, for what we're really trying to do now, and this is a big part of our vision moving forward, is to grow a robotic cloud lab network. To really enable creativity at transformative global scales. And so this is a figure from our Nature paper. One of the things that we imagine is having, for example, places at like MIT, Harvard, what we would call a supercore site. So a place where you could have a lot of equipment, mass specs, industrial robots, and liquid handlers, and so on. But then you could have regional nodes that could still be visited by students regionally, but still have robots. And a key part there, of course, is being able to share protocols, share best practices, share knowledge. All of that is done through this larger network vision. And so, what might we do with a network like this? This year, in addition to the 535 some students I mentioned, we've got more than 35 sites that want to be a part of our How to Grow network. So they want to be nodes in our network. And so this year, we're prototyping for the first time a pilot final project. And what we're doing is focusing on an application on pandemic preparedness and phage therapy. So for those of you that don't know, we're of course, in the midst of a real significant crisis related to antibiotic-resistant infections. And so, in particular for bacteria. And so one of the really promising ways to address this is using bacteriophages, which are these viruses that are very, very good in infecting bacteria, including bacteria that may be resistant to antibiotics. And so what the students have been doing is, through our protein design and DNA design classes, they've been learning all about how to design different types of proteins. And in particular, we're focusing on the proteins of these bacteriophages. And so the students all around the world are designing mutants of these bacteriophage proteins as a part of this phage therapy application. And then what we're doing for the very first time is enabling the synthesis and the expression of those proteins. And so we're doing that locally at MIT in partnership with Nuclera. They're a company that's made this amazing e-protein system. And so using digital microfluidics, we're expressing proteins made by MIT and Harvard students locally. And then we're distributing Opentrons robots to these sites that are all around the world. And so the idea here is that, and this is all using cell-free lysates, students around the world will then be able to actually synthesize their own proteins, their own mutant proteins that connect to this phage therapy application. And so we basically have this system now that's both methodologically innovative in that we're using creativity and DNA designs and protein designs from learners and students from all around the world. And then we're actually using this network of microfluidics and robots to then manufacture those proteins at scale and test them. And this, again, I think is a really exciting engine for creativity. And we're also, of course, doing new science. Many of these proteins, these particular bacteriophages that we're looking at, they haven't been studied, and these mutant that there hasn't been real experimental data gathered on these mutant proteins. So that's another area where we've got at least 2 levels of innovations on the methods and then also on the science. And all of this is made possible from an amazing set of industry partners. For those of you that are actually listening that might be excited to collaborate with us, we would love to collaborate with you. We're really grateful to Thermo Fisher, who's been helping us with the reagents and equipment. Twist Biosciences has been providing DNA synthesis. Nuclera, again, with these e-protein systems enabled by Biology DAO. And then Opentrons has been so incredible in partnering with us as an education partner to distribute their robots as a part of our How to Grow network. And right now, I mentioned, there's 35 robots sites that would love to have robots. And we're doing our first deployment of 10 robots right now to places in the US, Canada, Indonesia, India, Pakistan, Japan, Bhutan, throughout Europe, and so on. And so, again, for me, this model, I'm so excited about this moving forward. And there's all kinds of things you can imagine in terms of group projects that could be done. Just to give you an example of a specific learning module we're working on right now, I'm currently collaborating with Professor Ron Weiss from Biological Engineering Department at MIT and a Ph.D. student, Evan Holbrook. And and in the Weiss lab, they're working on this really cool project around neuromorphic circuits. So the idea is to take some of the computer science metaphors and models like neural networks and basically installing those into cells using genetic circuits so that the cells can actually edit their own DNA and basically train themselves on training data that's connected to genetic circuits that enable the measurement of different types of inputs and outputs and different types of weights that are associated with those inputs. And so this is a really amazing new kind of technology. And in a way, the cells in this context are basically like little platform tools. And so from a learning perspective and a creativity perspective, you can use these cells not just for medical applications, which is one, of course, big one, but you can use them for artistic applications or sensing behaviors or design applications. And so one of the visions here is to have students design their own neuromorphic circuits. This can be done in silico. They can be simulated and designed that way. And then using robotics, you can actually perform the transfections and circuit testing using the Cloud Lab network. Much like we're prototyping the synthesis of proteins all around the world, you can also prototype the performance of these neuromorphic circuits. And then the data that's generated through that work can then inform the next generation of circuit designs and simulations. And in a very similar way with the protein expression project that I mentioned, the phage therapy application, in a very similar way, you can take the data connected to the protein expression and filter that back into a next generation or a next round of simulation design, and then another round of expression and data collection. So this is a specific project we're working on to enable with this robotic cloud lab network. And lastly, I'll mention automation. So or biosecurity, excuse me. So, very grateful to my colleague Kevin Esvelt at the Media Lab and Lenny Justin. He's a student in How to Grow Almost Anything right now. Lenny, for his PhD, is really interested in robotics, but for How to Grow, for his final project, he's focused on a biosecurity application, right? So as we're distributing these robots around the world and starting to build out this network, one of the big things we're excited about is setting up biosecurity frameworks connected to that, right? So how do we look at protocols and potentially screen or look at different types of protocols to assess biosecurity risk? And then, of course, you can imagine using different types of AI and large language models to try and study and characterize these different protocols. And, of course, a huge part for us from an innovation side is enabling the sharing of protocols using a repository. I'll mention too, PyLab Robot. PyLab Robot is a really awesome open source framework for enabling the interoperability of robotics protocols. So you could have one protocol using PyLab Robot that can then be adaptable to Opentrons or Hamiltons or TCANs, depending on what your robotic format is. So really excited about PyLab Robot. And so that whole vision, is a big part of what we're really trying to grow here. So growing the robotics, growing the learning curriculum, growing global projects, growing biosecurity. All of that is a part of our vision here. And again, as I end, I'd so love to chat with anybody that is interested in collaborating. And we'd love, again, to talk with other industry partners as well to help realize this vision. Something we're really excited about. And I'm looking forward to answering any questions and towards hopefully collaborating with some of you that are listening today. So thank you all so very much. I'm going to exit out of my presentation right now and then stop sharing. Okay, and I think that was exactly 30 minutes. So I think we've got some time now for questions.
Sounds great. So thank you so much, David, for your very informative presentation. We will now go ahead and move on to the Q&A portion of the webinar. As a reminder, if you have a question you would like to ask, please do so now. Just submit a question in the Ask a Question box located at the bottom of your screen. All right, let's go ahead and get started. Okay, it looks like the first question we have here says, can you elaborate more on how this research can contribute to space exploration?
Great question. So we've had some really interesting conversations actually with folks that are in the space industry. So one big aspect that I didn't really talk about is reproducibility. So what could you do that happens here on Earth? And what could you do that happens out in space, right? So there's a really interesting way in which you could take robotics, put them actually in outer space, and then run the same protocols and see, what's different in zero G versus on Earth. And I showed that one example of that student who had that microfluidic wearable, and he actually tested that in zero G. So there's some stuff that we've done already there, but I think this in general, this kind of hybrid distance model, I mean, it literally could be hybrid distance to other to outer space and other planets. Even you could really imagine that. If you've got communications out to the moon or Mars or to a spacecraft, as long as you've got interoperable protocols and robots that are on Earth or in zero G or wherever else, there's no reason why you can't reproduce experiments, right? And that's the other kind of really exciting thing I think about working with robotics is the protocol sharing. We see this as well throughout different parts of planet Earth, right? So you could be in a really remote location as a learner and still program a robot that could be at MIT or on the International Space Station, right? So I think there's a lot of really exciting applications, and I had not totally thought about the anywhere to include outer space, but we absolutely should. I should zoom out from that picture and include the planet and some the galaxy too. That's awesome. I love that.
Perfect. All right, it looks like we have a few more here. The next
Sure.
one is, what advice can you provide upcoming students who wish to pursue this research?
That's an awesome question. So number one, join our class. We would love to have you. If you go to htgaa.org, you can find the website for the course. There's the whole curriculum is on there. You can see previous year's classes and students. We're going to do a revamp of the website at the end of the semester, but there's a lot of material and resources there. And basically the course, it's free, for our global audience. This is part of what's so exciting. You can be a listener, you can be a student anywhere around the world. And yeah, you can hop online and join us anywhere. There's an application process, but we basically take almost everybody. So we're really welcoming of learners, and every year, so we typically advertise it on social media. So I'm at David Sun Kong on all of the platforms, on Instagram, Twitter, X, etc. So I typically will post on it there. We're doing some more build out of our HTGAA social media, so look out for that in the future. But I always post about it, and you can see on my social media pages links to these to the applications. So we're in the middle of class right now. It's March 20th, and we're kind of I think like 6 weeks into our class right now. But we'll be doing more global project work over the summer, and there will be opportunities. So we'll advertise and talk about that on social media. And then definitely for next spring, I would really encourage anybody to come and join the course. And as I mentioned, once you join it, it's amazing because you get to just become a TA for life if you want. You're part of this learning community forever. And there's regional capacity that gets built out. If you're one of these regional nodes that's getting a robot, that's where resources are being sent to help build out a lab there, or built out a space there, there's leadership development and regional capacity building that can happen. So that's the other thing you could do too, is actually you could engage with one of our How to Grow nodes that are being set up. So we've got one in Tokyo, there's one in New York City at GenSpace. So there's a lot of places you could go regionally right now to go and plug in and learn. And actually, even right now, you could join in virtually and plug into our lectures. So there's actually quite a lot of ways. If you again kind of look at social media. At David Sun Kong, I share a lot of different ways to plug in there as well. So thanks so much for the question. And we'd love to have you. Anybody here listening, we'd so love to have you join our course.
All right, perfect. We have another here. It says, how many instruments, for example, LCMS, have you integrated into the workflow?
Oh man, I would love to integrate LCMS into the workflow. We have not integrated mass spec yet. We would love to do that. So again, there's two visions that relate to the architecture of the robotic network, right? One aspect of this is what we call, as describing as the supercore site. So what I would love to do is have, at MIT, a real integrated set of workflow that could be comparable to something like what Ginkgo Bioworks has. So for those of you that are maybe are familiar or not familiar, Ginkgo has got this really incredible architecture as it relates to the robotics. It's this big set of modular robots that can connect to each other, and you can kind of build in workflows with all kinds of different machines in there. And that I think would be, but of course they're using it for industrial purposes. For us, we're interested in this in a learning and a creativity capacity. And so there's ways in which I would love to integrate those types of tools here at MIT. For us, it's not just about that kind of mainframe, though. It's also about the distributed network, right? I think it's also really exciting to have lower cost robots. And again, really big shout outs to our collaborators and partners at Opentrons. It's really great to have a robot that you can go and play with and interact with in person, right? So that way you're learning about the synbio, but you're also learning about automation, which I honestly believe is really where the future of the life sciences and synbio is heading. One thing I didn't mention, and this is a huge part about this vision that I love, people have always been, and rightfully, concerned about biosecurity and things leaking out of labs and so on. With this type of robotics-enabled learning, all of the molecules stay at a supercore site like at MIT, or, again, right now, everything we're doing right now is basically biosafety level 1. So it's, kitchen biology is more dangerous than the type of bio that we typically work on. And what we want to do is basically have biosafety and security in a place like MIT, but then the innovation and the learning be radically decentralized to places all around the world with just an internet connection and a computer, right? So you could be anywhere and program robots at MIT and have a transformative learning experience. So I think, for us, it's like LCMS, I would love to do that at MIT as part of a bigger workflow. And that's something that we're working on making happen right now. I mean, that's something I'm really, really grateful for as well. I think, industry is really excited about this vision as well. And, we've been really, really grateful to our partners there.
All right, perfect. Okay, it looks like we have one final question that came in here. How do you
Great.
move a plate from the Opentrons to a plate reader?
Yeah, great question. So, yes, right now, a lot of the work does involve TA assistance, right? Like you still do need humans to intervene depending on how good your workflow and your automation pipeline is, right? There's some things you can do end to end, like some of our learning modules can be completely contained in the robot, and literally it's the robot executes it, you get the data out, and then you're done, which is amazing. Obviously, our goal is to do that for all of the systems if we can. But it's pretty cool. I think a lot of the latest robots are integrating a lot of different functionality in there. So there's quite a variety of assays you can start getting, assay data you can get out of these systems as well. There are robotic arms and things you can do, obviously, to kind of connect different machines, which we've been looking into and bringing into the system. But that's all a big part of this larger architecture that we're engineering right now. But great question.
All right, perfect. Well, thank you again, David Sung Kong, for your time today and for your very important research. Before we go, I would like to thank the audience for joining us today and for their very interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until March 20th, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.
Thank you so much.